Hi, my name is Amy Todd. I'm the owner of Zymology Labs, a consulting, training, and analytical testing lab for brewers and hard cider makers in Burlington, Vermont. Today I'm going to be showing you how to perform yeast cell counts based on weight. Performing cell counts and adjusting the amount of yeast you use based on your results is one of the best things you can do for the quality and consistency of your fermentations. Not adding enough yeast can lead to stressed out and overworked yeast. This causes off flavors and inconsistent fermentation rates. Adding too much yeast can also lead to off flavors and inconsistent fermentation rates. Cell counting is easy, and you can get everything that you need for under $500. Today I'm going to walk you through the steps and the equipment that you need. Let's get started.
Equipment needed for cell counting includes a microscope, scale, hemocytometer, 100 ml Erlenmeyer flask, spatula, Pasteur pipette and bulb, chem wipes, counter, parafilm, and a 0.01% solution of methylene blue. The more accurate your scale, the more accurate your results. Analytical scales can be quite expensive, so to keep costs down while still providing sensitive results, I like to use a triple beam balance. Typically, you'll start with a 1 to 100 dilution of your yeast slurry, but this will depend on how thick your yeast is. The dilution provides a manageable number of cells to count on the hemocytometer counting chamber. If you want to check the cell count of a fermenting beer, you can do that at a 1 to 1 dilution. To make a 1 to 100 dilution, you will weigh out 1 gram of yeast and then add 99 grams of water. First, you will need to zero your flask. To weigh out 1 gram of your yeast slurry, move the front weight to the 1 gram position. Then use your slurry to rebalance the scale. Once you have added 1 gram of yeast, move the 1 gram weight back to 0. Now move the 100 gram weight into place. This will give you a total of 100 grams, 1 gram of yeast, 99 grams of water. Once you have your 1 to 100 dilution, make sure you give it a shake every time you're about to take a sample because your yeast cells can settle out. Now it's time to load your hemocytometer. Make sure your hemocytometer has a special hemocytometer cover slip. This is a specific size and weight that will help you fill the right amount into your cell. There are 2 tiny chambers in the hemocytometer with a known volume. This is how you are able to calculate how much yeast is in 1 ml of your slurry. I like to use hemocytometers with these little V markings here. It makes loading your hemocytometer a lot easier. Pull up a sample of your slurry, expel a few drops, and then you're going to want to get a drop on the tip of your pipette and touch it to the side of your slide. The V will wick up your slurry. This takes a little bit of practice, but once you do it a few times, it will become quick. Now it's time to count your yeast. You're going to start by loading your slide onto your microscope on the lowest objective. Once you have found your yeast and have them in focus, move to the next objective. Refocus and move to the 40x. This is where you're going to ultimately be counting your yeast. From here, you'll be looking for a grid. You can follow the triple lines to find 4 by 4 squares that comprise of 25 squares that make a larger 5 by 5 grid. You're going to be counting the 5 corner squares and the center. Count the cells either touching the top and the right or the bottom and the left of the sides. This is so you don't count the same cells more than once. I like to perform my viability tests separate from my hemocytometer count. To do this, I take 1 ml of my yeast slurry and mix it with 1 ml of my methylene blue solution. I combine it in a small dish, let it sit for 2 minutes so that the stain is able to do its work, and then I'll add a drop onto a separate slide
and use that to count. I always mix up my yeast right before use. Use a transfer pipette and a separate one for my methylene blue solution because I don't want to introduce any yeast cells into my stock solution. I set my timer for 2 minutes and then count how many blue cells I can find.
Check out www.zymologylabs.com to check out worksheets on cell counting along with other resources. Thanks for watching.